Next, Homo neanderthalensis. This species lived in Europe and Asia from around 400,000 to 40,000 years ago and had a larger brain than Homo erectus, as well as more advanced tools and complex social structures. Next, Homo sapiens. This species evolved in Africa around 300,000 years ago and gradually spread out across the globe, replacing other hominid species along the way. The evolution of Homo sapiens was driven by a combination of genetic mutations and environmental factors such as changes in climate and landscape. These factors created selective pressures that favored individuals with certain traits such as larger brains and increased social skills. Then evolution of horses. The evolution of horses has a much longer story spanning over 50 million years, and horses belong to the order Perissodactyla, which also includes rhinoceros and tapirs. The earliest known ancestor of horses was a small, dog-sized animal called Hyracotherium, which lived around 55 million years ago. Over time, horses evolved to become larger and more specialized for running, with longer legs and a single elongated toe. The evolution of horses can be broken down into several major stages like Eohippus. This early horse ancestor lived around 55 million years ago and had 4 toes on their front feet and 3 toes on their back feet. Then Mesohippus. The species lived around 40 to 30 million years ago and had longer legs and a more elongated skull than Eohippus. Then Merychippus. Species lived around 17 to 11 million years ago and had evolved into a true horse with a single elongated toe on each foot and more advanced teeth for grazing on grass, like Merychippus. Then Pliohippus. The species lived around 5 million years ago and was the first to have a fully developed single toe and a large brain. Then Equus. This is the genus to which modern horses belong, and it includes species such as the domestic horse Equus caballus and the wild mustang Equus ferus. The evolution of horses was driven by selective pressures related to their environment, such as the need to outrun predators and access new food sources. Additionally, genetic mutations and interbreeding with others is also possible. Then island ecology. Island ecology is the study of ecological processes and patterns that occur on islands, which are isolated and often unique habitats. This process can include speciation, extinction, colonization, and adaptation. One of the most important aspects of island ecology is the study of island biogeography, which was first proposed by Robert MacArthur and E.O. Wilson in 1960s. This theory explains the patterns of species richness and diversity on islands as a balance between immigration and extinction rates. The theory suggests that larger islands will have higher species richness and that the islands of a distance of an island from the mainland will affect the rate of colonization and extinction. Other important concepts in island ecology include the study of island food webs, island biotas, and island biogeography. The unique conditions of island habitat also provide an ideal setting for the study of evolutionary processes and adaptive radiation, where one ancestral species gives rise to a variety of different species, each adapted to a different ecological niche. One of the most famous examples of island ecology is the Galapagos Islands, which were instrumental in the development of Charles Darwin's theory of evolution. The unique and isolated conditions of the Galapagos allowed for the development of many endemic species, which were the subject of Darwin's studies. Another well-known example of island ecology is Hawaiian Islands, which are home to a diverse range of species and unique ecosystems. Island ecology is important for understanding the complex interactions between species and their environments, as well as the mechanisms that drive the evolution of new species. This knowledge can be applied to the conservation and management of island ecosystems, which are often threatened by human activities such as habitat destruction and introduction of invasive species. Next, plant biology, floral diagram. So, floral diagram and general morphology are important aspects of plant biology that help in the identification, classification, and understanding of the evolutionary relationship of plants. Floral diagram is a graphical representation of a flower structure of a plant. It provides information about the arrangement, number, and structure of the various floral parts. In a floral diagram, the floral parts are represented by symbols, and the position and the relationship of these symbols indicate the relative position and relationship of the corresponding floral parts. General morphology. General morphology refers to the physical appearance and structure of plants. The general morphology of plants includes the study of various plant organs such as roots, stems, leaves, and flowers. Each of these organs has specific structures and functions that enable the plant to carry out various physiological processes. The importance of floral diagram and general morphology. Floral diagrams and general morphology are important tools for plant identification, classification, and evolutionary studies. They provide a visual representation of the unique characteristics of different plant species, which can help in distinguishing between different species. By studying the general morphology of plants, we can also understand the adaptations that plants have developed to cope with different environmental conditions. For example, the structure of the root system can provide information about a plant's ability to tolerate drought or nutrient-poor soils. Floral diagrams and general morphology can be used to study a wide range of plant species, from simple plants like mosses and ferns to complex angiosperms. For example, in angiosperms, the floral diagram can provide information about the position and number of floral parts such as sepals, petals, stamens, and carpels. By studying the general morphology of plants, we can also understand the adaptations that have evolved in different plant species. For example, the presence of specialized structures like succulent leaves in cacti help them to store water in arid environments. Next, brasinosteroid signaling. Brasinosteroids (BRs) are a class of plant hormones that play a key role in regulating various aspects of plant growth and development, including cell elongation, muscular differentiation, reproductive organ development, and stress response. The signal transduction pathway of BR is a complex process that involves multiple components, including receptor kinases, co-receptors, transcription factors, and other regulatory proteins. The BR signaling pathway starts with the binding of BRs to the plasma membrane localized to the receptor kinase. Then BRI1, which triggers the activation of BRI1 and the subsequent requirement for its co-receptor, BR1 associated receptor kinase, BAK1. The binding of BRI1 to BR induces a conformational change that leads to the autophosphorylation and activation of BRI1 as well as the phosphorylation of its downstream targets. The activated BRI1 BAK1 complex phosphorylates several downstream proteins, including the GSK3-like kinase, BIN2, and the transcription factor BZR1. BIN2 is a negative regulator of the BR signaling pathway as it phosphorylates and inactivates the transcription factors BZR1 and BZR2, which are key components of the BR-mediated gene expression network. The inactivation of BIN2 by the BRI1 BAK1 complex leads to the dephosphorylation and activation of BZR1 and BZR2, which then enter the nucleus and regulate the expression of BR responsive genes by binding to the promoters of target genes. BZR1 and BZR2 interact with several transcription factors, including members of the basic helix loop helix family, to regulate the expression of downstream genes that are involved in various physiological processes. The BR signaling pathway also intersects with other signaling pathways such as auxin signaling pathway to coordinate the growth and development of plants. For example, the BRs and auxins synergistically promote cell elongation by regulating the expression of genes involved in cell wall biosynthesis and modification. The importance of BR signaling in plant growth and development is underscored by the fact that mutations in BR biosynthesis or signaling genes often result in severe developmental defects such as dwarfism, reduced fertility, and altered response to environmental stimuli. In addition, the manipulation of BR signaling has been shown to improve crop yields and increase stress tolerance in various plant species, making BRs a promising target for crop improvement strategies. Hormone function. Plant hormones functions. Plants produce various hormones that regulate growth, development, and response to environmental stimuli. Auxins. These hormones promote cell elongation, stimulate cell division, and affect the orientation of cell expansion. They also play a role in apical dominance, tropisms, and root development. Gibberellins. These hormones promote stem elongation, seed germination, and flowering. They also help to break seed dormancy, stimulate cell division and differentiation, and regulate fruit development. Then cytokines. The hormones promote cell division and differentiation, delay senescence, and help to regulate apical dominance. They also play a role in shoot development to control the balance between shoot and root growth. Then abscisic acid. This hormone is involved in stress response such as start, cold, and salinity, and promotes seed dormancy and inhibits growth during adverse conditions. Next, ethylene. It is a hormone regulating fruit ripening, promote leaf and flower senescence, and coordinates plant response to biotic and abiotic stress. It also plays a role in gravitropism, and apical hook formation in seedlings. Then brasinosteroids. These hormones are involved in cell elongation and division, differentiation, and stress response. They also promote flowering, seed development, and root growth. Next, we will be learning tomorrow. Thank you for listening.